Hello, welcome to Biology, the scientific study of life. The life cycle of Rhizobos, stonifer, blackbread mold is fairly typical of zygomycetes. Horizontal hyphae spread out over the food, penetrate it, and absorb nutrients. The hyphae are synustic with septa found only where reproductive cells are formed. In the sexual phase, bulbous black sporangia develop at the tips of upright hyphae. Within each sporangium, hundreds of haploid spores develop and are dispersed through the air. Spores that happen to land and moist food germinate, growing into new mycela. Some zygomycetes, such as Pilopols, can actually aim their sporangia toward conditions associated with good food sources. If environmental conditions deteriorate, for instance, if the mold consumes all food, Rhizobos may reproduce sexually. The parent in sexual union are mycelia of different mating types that possess different chemical markers. Although they may appear identical, plasmogamy produces a sturdy structure called a zygosporangium, in which karyotogamy and then meiosis occur. Note that while a zygosporangium represents the zygote's second generation stage in the life cycle, it is not a zygote in the usual sense, that is, a cell with one diploid nucleus. Rather, a zygosporangium is a multinucleate structure, first heterokaryotic with many haploid nuclei from the two parents, then with many diploid nuclei after karyotogamy. Zygosporangia are resistant to freezing and drying and are metabolically inactive. When conditions improve, a zygosporangium undergoes meiosis, germinates into a sporangium, and releases genetically diverse haploid spores that may colonize a new substrate. Microsporangia. Microsporangia are unicellular parasites of animals and protists. Notice this figure, et voila. They are often used in the biological control of insect pests. While microsporangia do not normally infect humans, they do pose a risk to people with AIDS and other immune compromised conditions. In many ways, microsporangia are unlike most other eukaryotes. They do not have conventional mitochondria. For example, as a result, microsporangia have been something of a taxonomic mystery, thought by some researchers to be an ancient, deep-branching lineage of eukaryotes in recent years. However, it has become clear the microsporangia are not primitive eukaryotes, but rather are highly derived parasites. In 22, Bryony Williams and colleagues at a Natural History Museum in London discovered that microsporangia actually have tiny organelles derived from mitochondria. Meanwhile, molecular comparisons have revealed that microsporangia are more closely related to fungi than to any other eukaryotes. A 23 analysis provided some data suggesting that microsporangia should be classified as zygomycetes. Microsporangia demonstrate the extraordinary changes that can occur as organisms adapt to a parasitic lifestyle. In their case, they lost almost all resemblance to their fungi relatives. Glimoromycetes, the Glimoromycetes fungi assigned to the phylum Glimoromycetes were formerly thought to be zygomycetes. By analysis of hundreds of fungi genomes indicate that Glimoromycetes from a separate clade monophyletic group. In spite of their small numbers, only 106 species have been identified to date. The glymeromycetes are an ecological significant group. All glymeromycetes formed a distinct type of mycorrhiza called arbosicular mycorrhiza. The tips of the hyphae that push into plant root cells branch into tiny tree-like structures known as bisicules. About 20% or 90% of all plants have such symbiotic partnerships with glymeromycetes. Notice this figure, et voila. Basidiomycetes. Let's talk about the basidiomycetes. Approximately 30,000 fungi, including mushrooms and shell fungi, are called basidiomycetes and are classified in the phylum Basidiomycetes or Basidiomycota. The phylum also includes molds, mutualists, that form mycorrhiza, and two groups of particularly destructive plant parasites, the rusts and smuts. The name of the phylum derives from the basidium, a cell in which a transient diploid stage occurs during the fungi life cycle. The club-like shape of the basidium also gives rise to the common name club fungi. Basidiomycetes are important decomposers of wood and other plant material. Of all the fungi, the subaerobic basidiomycetes are the best at composing the complex polymer lignin and abundant component of wood. Many shell fungi break down the wood of weak or damaged trees and continue to decompose the wood after the tree dies. The life cycle of basidiomycetes usually includes a long-lived dikaryotic mycelium, as in zygomycetes. This extended dikaryotic stage provides opportunities for many genetic recombinations to occur, in effect multiplying the result of a single mate periodically in response to environmental stimuli. In mycelium reproduces sexually by producing elaborate fruiting bodies called basidiocarps. A mushroom is a female example of basidiocarps. By concentrating growth in the hyphae of mushroom is a basidiomycete. Mycelium can quickly accrete its fruiting structures in just a few hours. A mushroom pops up as it absorbs water and cytoplasm streams in form the dikaryotic mycelium. By this process, a ring of mushrooms, popularly called a fairy ring, may appear on a lawn literally overnight. Notice that. The mycelium below the fairy ring expands outwards at a rate of about 30 centimeters per year, decomposing organic matter in the soil as it grows. Some giant fairy rings may be centuries old. The numerous basidia in a basidiocarp are the sources of sexual spores called basidiospores. The mushroom's cup supports and protects a large surface area of basidia or on gels. A common store-bought white mushroom has a gel surface area of about 200 centimeters square and may release a billion basidiospores, which drop from the bottom of the cap and are blown away. Asexual reproduction is much less common in basidiomycetes than in zygomycetes. Okay.